only a brief introduction. It's great that Nancy Kanwisher with us today. Nancy is a professor of cognitive neuroscience here in the department, brain cognitive science, and a member of the center of CBMM. She's also a world leader in her area, which I would say her focus is a broad focus on the mechanisms and representations in the brain, in vision, but also outside vision, as we will do a little bit, and also the exciting area of development and plasticity, both in normal development and in disease. And, you could see from the papers that you sent some of the recent and very exciting, interesting direction which we talked about. And also, if you had an opportunity to look at the overview and perspectives, this year is 20 years since the publication of the paper on the face areas, the face area in the brain of the face, with Josh Bennett and Martin Shunt on the right.
Arvidsson, yeah.
Co-authors. And this was an opportunity to also look back and have a broad perspective on the area, intentions and progress and promising directions. So look at it and should enjoy it, and please use this opportunity, to listen to some specific scientific material, but also to get some broad perspective on the field. So please do not hesitate, and if you have some questions and issues you want to discuss, it's a good opportunity to go.
Great. Thank you. Hi. Yeah, so as Shimon said, I'm going to do like very broad, big picture here. And I'm trying to not assume too much background because I know there's quite a range of backgrounds, but I'll try to be sensitive. There are things that maybe Shimon can help me. If there are things that he feels people are totally on top of, you can just butt in and tell me to skip that or something. Yeah, great. So I thought this is sort of planned outline. I'll start with just some general perspectives on what kind of architectures we might want for intelligence. And as I'll argue, a long debate in many fields has been to what degree do we want special purpose machinery versus general purpose cognitive machinery. And that's played out through the history of psychology and neuroscience and arguably computer science. We have this all coming up in weird orders. Doesn't matter. Okay. Then I'll talk very generally about how we can use functional MRI to look at the organization of the human brain. And then I'll talk about some very specific work on particular specializations in the human brain for particular mental functions. And in the last part, I'll talk about some of the ongoing work on what you might think of as the neural basis of the cognitive core. That's the CBMM lingo for this module 3 that Josh Tenenbaum and I are directing. So that's the agenda. And I thought I'd start with some going way back in the history of psychology to the work of Spearman, who was very interested in intelligence and what it meant. And he published an article in 1904 in the American Journal of Psychology. And it was sandwiched between a discussion of the soul and an account of the psychology of the English sparrow, just to set the kind of times. And what Spearman did was an extremely low-tech but a kind of deep question. He went into schools and he tested children on a whole bunch of different tasks. He tested them on, he got measures of their academic ability from exam grades in a whole bunch of different subjects. He measured their ability to discriminate different kinds of information, like the pitch of tones or weights of different objects or the brightness of a light. And from all of these different abilities, he looked at the question of whether they were correlated within an individual. In other words, so he asked in particular, if you found a kid who, say, tested very well on math exams, would they be better at discriminating the brightness of two patches or the pitch of two tones? So what do you guys think? Would you think those things would be correlated or not? Everybody get the question? So generally, if you're good at one thing, are you good at other things as well? And I'm choosing a kind of ludicrously broad stretch between, say, math ability and perceptual ability to discriminate brightness and tones.
I
Yeah.
guess if it's using the same area of the brain, I guess it makes sense for recorded. And I guess I have a hard time believing the argument.
Right. Okay, I think that's a good answer. Any other? That's certainly my intuition. Any other perspectives? Well, the weird thing that Spearman found was that, in fact, most tasks are correlated within an individual, even widely different things that you would never expect to be recruiting common brain machinery. And so he found those who do well at school are the same as those who are good at sensory discrimination, to a obviously not perfect extent, but to a surprising degree. Everything was pretty much correlated with everything else. And so I just want to point out that it didn't have to be that way. And this is a potentially kind of profound insight about the organization of intelligence. And certainly that's how Spearman took it. What he inferred from this is that there's a general factor which he called G, and it's this line of work that led to intelligence testing and the whole idea of intelligence as an individual difference variable describing people. And in this country, we're mostly very squeamish about this idea. It seems very politically incorrect and very unsettling to think that some people are endowed with just generally better brains than others. In England, they're not uptight about that at all. They're very comfortable with that idea. They test people early on, decide whether they have good brains or not, and determine the rest of their lives. I don't think it's as bad as it used to be, but there's certainly that tradition. And so what I want to point out is, yes, I share all those typically American sentiments. I hate the idea of fixing people's possibilities and tracking them and so forth. Nonetheless, if we want to understand how brains are organized, this question's a very interesting one. And the fact that there does seem to be this G factor, there does seem to be this correlation across widely different tasks, does seem to be capturing something about the architecture of human intelligence. I'll also point out, though, that while Spearman is best known for G and for the whole enterprise that it launched, he also talked about S factors, the specialized factors. So what he said was, if you look across all these correlations across tasks, you find that the first thing you see, like the first principal component, is G, that explains lots of variance across individuals. But on top of that, certain pairs of tasks, or certain tasks are less correlated with G than others. And so in addition to this general idea that brains have a general purpose mechanism, he also argued that there were some tasks that just didn't fit that well with G. They were all correlated, but less. Some of the ones that were particularly less correlated with G were ability to learn Greek. Ability to learn Greek was highly correlated with G, but musical ability was less so. Okay, so there's sort of both general and specialized machinery. And as you might be guessing, I think this is kind of, in many ways, foreshadows what happened in the history of cognitive neuroscience. Okay, so this, and actually cognitive science before that. So cognitive science, especially of the 70s and 80s, a big question that was much debated then is the degree to which human intelligence is composed of special purpose machinery versus general machinery. Right? And so this was much debated by Fodor and Chomsky and people like that, looking at how human minds work using different methods from Spearman, but similar in idea. And so one of the analogies that was put forth by Leda Cosmides long ago, based on sort of evolutionary considerations, but you don't need to buy into that to raise this question, is whether we should think about the human mind as kind of like a Swiss Army knife with lots of very specific devices, computational devices that solve different kinds of problems. Or whether we should think of human intelligence as more kind of general purpose without being specialized for tasks in particular. Yes? The environment. Say more what you mean. Are you saying, is the structure of the task we have to solve compartmentalized? Yeah, I think that's a great question. Absolutely, and one can ask that in all kinds of different ways. But for example, Marr argued in his magnificent 1980 book on vision, where if you haven't read the first few chapters, you should drop everything and do that. It's more important than anything else you might read. It's beautifully written and really deep. And one of the things he points out is that the problem of vision has all of these different components that seem computationally very different from each other. Like if you think about the problem in color vision, in color vision, you're trying to figure out what is this thing made of, or what is a property of this material, the color of this, that doesn't have any color. Nothing here has any color. What is the color of this thing, right? And the problem there is that the light coming from that thing hitting your eyes is a property not only of the material of this thing, but of the incident light hitting it. And so that is like one little piece of a problem of vision. Color vision, what is a fundamental problem? That's a fundamental problem. That's a different kind of thing from solving problems of, say, stereo vision, 3D vision, using the different perspectives of the two eyes. And that in turn feels like a pretty different thing from motion perception or shape perception or face recognition, right? And so Marr argued that whenever you have a problem that is inherently compartmentalized, I'm sort of reading in, this is a little bit more my reading in, but you can, with some poetic license, you can imagine Marr saying, where the problem itself breaks down into natural components, that's where you might want computational machinery that has specialized bets. Did I read too much into Marr, or is that?
No, anyway, I think it's part of this important point because people who sort of endorse the Swiss knife metaphor often treat it as just a quirk of evolution. They say, well, evolution went this way and another way and came up with this solution. But there may be something deeper that the optimal solution to certain problems is
Exactly. Yes. I think that's a much more interesting version of the Swiss Army knife metaphor, even though the people who came up with it are very invested in evolution as telling us which of these things should matter. In fact, I'll argue in a little bit that some of the special purpose mechanisms that are, well, at least one special purpose mechanism in the human brain that's highly, highly specialized cannot possibly be the product of natural selection. So for at least one, there's evidence. Okay, so this question has just raged in the field in various versions for a very long time. And it's raged even longer in neuroscience, where you have actually the infamous Franz Joseph Gall, but also he was a very accomplished neuroanatomist, Austrian neuroanatomist, who went around Europe giving lectures on the human brain and doing public dissections. And there were lots of bitter battles between him and his rivals about his basic idea that there are special purpose mechanisms in the brain. But no one disputed his neuroanatomical prowess. But so Gall, had this big idea that the brain is composed of what he called distinct faculties. And that was a big idea. But then he wanted to know what those faculties were and where they lived in the brain. And this being the late 1700s, he didn't have many options, and so he invented the best he could. He invented the method of phrenology, which is kind of used as classic pseudoscience. The idea was if a person had a particularly strong specific ability, then the part of the brain that engaged in that ability would be larger, and maybe you could discover this by feeling bumps on the skull. Maybe. Anyway, from this he came up with 27 mental faculties. My favorites are amativeness, filial piety, and veneration. Needless to say, we don't think these things are localized in particular regions of the brain today. And this was kind of a fringe view at the time, and certainly a fringe method. And at the time, he was much maligned by other scientists of the day, like Florence, who's often credited with launching experimental neuroscience. He did lesions in pigeons and rabbits and other animals, and tried to figure out whether you would get very specific deficits or very just general, just generally stupid pigeons and rabbits, or pigeons and rabbits who would lose particular abilities. And he argued for the former view, that pretty much whatever bit of brain you took out, you would just get a general across-the-board decrement in abilities. So from that, he reasoned, All sensory and volitional faculties exist in the cerebral hemispheres and must be regarded as occupying concurrently the same seat in those structures, right? So all mental functions are on top of each other in the brain. That's the idea. So he was sort of the more respectable scientist and the kind of, the better view, the more respectable view, until Paul Broca came along and stood up in front of the Anthropology Society of Paris in 1861 and announced that the left frontal lobe is the seat of speech on the basis of this patient, Tan, so named because Tan was all he could say after sustaining this nasty lesion to his left frontal lobe. Okay? And so once Broca argued that speech had this special purpose brain machinery revealed by this selective deficit, that became sort of an acceptable topic for debate, and this debate continued on and continues to the current day. So I'd say that currently there's very widespread agreement that basic sensory functions, visual cortex is back here, auditory cortex is up here, motor cortex is up here. Nobody's really seriously debated that those things do more or less what they do. Actually, even that's not true. There's a whole line of thinking that says that visual cortex isn't really about vision, it's about spatial stuff, and if you're born blind, you can use tactile information and process spatial information in primary visual cortex. So anyway, but the standard view is that those basic sensory and motor processes do get specialized brain machinery, but higher level perception and cognition, that's where all the fights ensue. Okay. So that's a perspective from neuroscience. This is a screen grab I took of an article that came out in the New York Times a month ago. And most of you computer scientists here will be able to evaluate the claims in this article better than I can, but I found it particularly interesting and relevant for the topic of today. So they argue that now some of the world's largest tech companies are taking a cue from biology. They're rethinking the very nature of computers, building machines that look more like the human brain, where a central brainstem oversees the nervous system and offloads particular tasks like hearing and seeing to the surrounding cortex. Okay, so this article makes a whole bunch of claims that basically, I don't know, it's too boring to read through, but the basic idea is that computer science is undergoing a revolution very recently, whereas instead of making more and more powerful CPUs, which has sort of been the main way to advance the field for decades, that's plateauing. And so instead, what's happening is people are making more additional special purpose chips that work in concert with those general purpose machines. And that in some cases, they're building specialty chips specifically to do what other chips have learned and so forth. And so I think this is a possible parallel to this view of the brain. If this is really what's going on, whether or not it's actually been informed by neuroscience, or if it's just what computer scientists would have done anyway, I don't know. To me, what's interesting is to the degree that this seems to be a developing solution to big computational problems in computer science, then it's very interesting to, if indeed the brain has come up with the same solution. Okay, so what can we learn from the human brain? Again, popping up in the wrong order. So let's take this question of the functional organization of the human brain. So I want to point out that the advances in understanding the organization of the human brain are really quite recent, right? So on the far left over here is a picture of the organization of the human brain published in 1957 by the famous Canadian neurosurgeon Wilder Penfield. And he'd spent a lifetime operating on human brains and electrically stimulating parts of the human brain while doing it to map out functions of the brain. Okay, and I'll show you shortly what can happen when you electrically stimulate the human brain. It's quite spectacular what you can see. So after a lifetime of doing that, he publishes this map of the human brain over there. And the main point, even though you probably can't read it, is there's just not much detail there, right? So despite all of this work, as early as 1957, we just basically didn't have that much information about how the human brain was organized. And if you cut ahead to 1990, up here, just before the invention of functional MRI, the picture was not much better, right? So the black outlines are my very impressionistic picture of where primary sensory and motor functions are. As I mentioned, those have been known for a long time. In addition, based on studies of patients with brain damage, like patient 10 who lost speech, you could make very loose arguments that very roughly in those big blurry locations, if you had damage in those locations, you might have deficits in language or attention or face recognition, right? Is that a question? Yeah.
Yes, sir, but what are the black?
The black ovals are just roughly the location of primary regions, like primary vision, primary visual cortex is back here, primary auditory cortex, primary motor regions. Yeah, and I put those in just because nobody disputes those, and those have been known a long time. And as I mentioned, the real question in this whole organization of the functional architecture of the brain is, what about all the higher level stuff? Is there a question there? No. Okay, so that's a picture right before functional MRI. And then functional MRI comes along, and now we have this extremely rich picture where there are dozens of regions in the brain for which we can assign a pretty specific mental function to that brain region. So what I want to do today, and I think this is worth knowing, I think it's interesting, I think it's important. So what I want to do today is consider what this all means, right? And where we can take it, what exactly we do and don't know, what functional MRI can reveal about it, and ultimately I think the agenda is to consider what this tells us about human intelligence and what it tells us about design of computer intelligence. Is there a question back here?
How distinct the regions are, or in other words, like what is the ability of the brain to compensate the lack of motor decision considering?
Okay, those two good questions, but two different questions. How distinct, right? So I've drawn them as nice little ovals. I blurred the edges slightly, but not very much. Nothing is that clean in the brain, right? So things are sloppy and messy. But if you, for example, take the standard way of defining the fusiform face area, and I'll describe that in a minute if anybody would find that. It's about 0.5 centimeter across by the standard way people find it, right? And then if you ask, okay, how does its functional signature drop off as you move millimeters outside that edge? You lose that face specificity that's defining of that region by the time you're about 4 millimeters outside its standardly defined edge. Which is, depending on your perspective, it's not like a butte, it's like some blurry thing like this, but it's not like the whole brain. It's not blurry all over the place, okay? And I think that's pretty typical. And some of the regions that nobody disputes as like things in the brain, like visual motion area M was studied first in monkeys going back many decades, and much is known about it in great kind of, really magnificent computational detail. Where you can record from hundreds of individual neurons and really characterize the neural representation with spectacular precision in a way you can't with functional MRI. Nonetheless, if you look at the spatial, sharpness of visual motion area MT, it's scandalous. I mean, motion selectivity extends in a lesser degree for centimeters beyond the typical thing, and nobody disputes that that's still a thing worth considering. So bottom line, the edges are not super sharp, but point two, maybe that doesn't matter so much. I mean, I think really ultimately what we care about is this thing functioning as some kind of computational system? And that will have more to do with whether there's a coherent subset of neurons that are talking to each other, doing similar things, getting common input, and maybe they're a little bit interleaved with other neurons around their edges, making a kind of blurry picture here. But there still may be something relatively discrete underlying it. You asked something else. Oh, can they move over after brain damage? Yeah? Yeah, so that's a very good but different question, right? And I would say to a first approximation for most of these, if you had damage to any of these regions in the first few years of life, you would probably be able to reorganize your brain and develop many of those functions elsewhere. It's not certain for all of them, but for many of them, you would probably be able to. But if you had damage to those regions now, mostly you're screwed. Some of these, there are compensatory mechanisms. You can use alternate computational strategies to solve the same problem without the special purpose machinery, and so it's not like you can't solve those problems at all. But to a striking degree, most of these functions are not relearnable if you lose those regions in the native way you had before, if you lose them in adulthood. Yeah.
Are any there are any other locations or some regions dependent on the location of the mass?
Yeah, it's a great question. What does next door mean in the brain? Is that sort of, yeah, in the cortex. And I should also say, when I say brain, I mean cortex. I'm a cortical chauvinist. There's no good reason for it. It's just what I do, and life is short. And so I'm focusing on the cortex, the outside stuff. There's lots of fascinating stuff down in the basement, but I'm not the person to tell you about that. Are there reasons why things are next door to each other? I think that's a great question. I would love to know the answer to that. I have not seen anything sensible written about it. And certainly if you look at some of these functions, it's really hard to tell how that would work. So as I'll argue in a bit, lots of stuff up in here is involved in various kinds of social functions, and that seems quite distinctive. Many different kinds of social functions, but they're all kind of social, right, in very different ways. So maybe it's meaningful that they're nearby, maybe it's not. The best guess about why that might matter is that there are reasons to think that reducing wiring length is an important constraint in design of brains. And so if you think that two different regions that may be doing slightly different things need to talk to each other, you'd want to put them nearby to minimize wiring length. But they're also, in addition to regions like this where you could tell a sort of BS story about, oh, social stuff is up there, right? There are also things that make no apparent sense. Like down here on the bottom of the brain, very systematically and very interestingly, the exact same configuration is found in monkey brains. So it's a little hard to tell in this map here, but there's a band of face selective regions right there. This is the bottom of the brain, back of the head, everyone oriented. There's a face selective regions in the dark purple here, a band of color selective regions right in the middle, right next door, and a band of place selective regions right there. And maybe you guys, you computer scientists, can come up with some story about why faces should be next to color, should be next to places, but it doesn't do anything for me. Nonetheless, that systematic organization on the bottom of the brain, like right in the bottom of my temporal lobes here, faces, color, places, is quite clearly mirrored in monkey brains way out on the lateral surface, where on the lateral surface it goes faces, color, places out there. So maybe there's a reason for it. That would be cool. This seems like a case where, if I had to guess, I would guess that's just kind of an evolutionary thing. It just happened early on and got inherited and pushed around. But who knows? Yeah.
The question of how distinct these regions are, isn't that also a question of how do we define functional organization in the first place?
Yeah.
Like, why should we not break speed perception further down into stuff?
Absolutely. Absolutely. So when I say with this picture that there are bits of brain for which we can ascribe a pretty specific function, at some level that statement has no content unless exactly how specific and what kind of specific, right? So I will say what I mean by that, but you're absolutely right. And so I think it's a very big open question about what actually is the grain of the organization. And almost all of the work in humans is necessarily looking at a particular grain just based on the resolution methods. And so there could be spectacularly more precise functional organization at a much finer grain in humans, and we just wouldn't know it because we don't have the methods to see it, right? However, as I just argued, I think that this whole system in here is very homologous between humans and monkeys. And that's good news because it means that the limitations of the currently available methods in humans can be complemented by methods in monkeys where you can really do much, much more. You can record from individual neurons, then you can record from hundreds of individual neurons. While the monkey looks at thousands of different stimuli, you can really analyze the neural representation of those stimuli, the timing of those computations, the connectivity. Don't get me started. I'm in a really depressive mood these days because we can't do any of those things with humans. Right? So all we can do with humans is take big chunks of that system and kind of average across it with our big blurry MRI methods. Nonetheless, impoverished as the methods in humans are, I still think this picture is worth having, right? Even if that's all we have as a sketch of where to go from there. Yeah? Did I get off the track or did I? I forget. Okay, sorry. Okay, so I could like 5 minutes and say what exactly, how does a bare bones, simple functional imaging experiment go? Or maybe you're all familiar with that. Would anybody like me to say how you define a phase selective region in the brain, or is that?
It would be useful.
Well, okay. So those of you who've heard this before, you can read your email. I won't be insulted. Oh, some basics of functional. Okay, so functional MRI uses the same old machines as regular MRI. And the key advance that happened in the early 90s was some smart physicists figured out how to get these machines to take the pictures fast. And second of all, how to take pictures with MRI machines that show not just the density of tissue, but neural activation. Okay? And the idea behind the neural activation is that brains are like muscles. If a bunch of neurons in a particular region of the brain fire, that's metabolically expensive, just like tensing your muscles or running. And just as when you run, you need to send more blood flow to your leg muscles to supply those muscle cells. If you use a bunch of neurons in here and they fire a lot, you need to send more blood to those neurons to supply them metabolically. And thankfully, blood control to the brain is quite local. Thankfully, because if that weren't true, this method wouldn't work. And so what functional MRI does is detect local differences in oxygenated and deoxygenated hemoglobin. They're magnetically different in a way that an MRI machine can detect, and that's the basic signal. The reason all of that is relevant is that this basic principle behind functional MRI reveals a bunch of limitations in the method. So let me just mention a few of those. So because the method is basically a blood flow method, so a bunch of neurons, imagine a bunch of neurons in my primary visual cortex back here. You flash a light for a checkerboard for 100 milliseconds. We know from other work that about 80 milliseconds, maybe 70 milliseconds, neurons in my V1 will fire. And then they'll shut off. The checkerboard's off, the thing is done, they fire, boom, they're done. Everything's over in a few hundred milliseconds. Okay? So all of that happens way over here on this timeline. I mean, right near the zero line. That's the MRI response. 6 seconds later, blood flow changes in that region. I mean, gradually ramping up and peaking 6 seconds after that neural activity. Okay? So that's a drag if you're interested in how information is processed at a fine temporal grain, which if you're interested in the details of how the computation unfolds, you absolutely want to know things unfold on the order of at least tens of milliseconds. And sadly, we're off by a couple orders of magnitude in our ability to resolve those little steps of a computation. That's just life. Yeah? Okay. That's what I just said. Okay. So because the basic signal is based on blood flow, not only do we not have this fine temporal resolution, do we not have fine temporal resolution? But the spatial resolution is also not quite what we'd want. It's by far better than anything else available in humans. From the point of view of a systems neuroscientist, it's a scandal. So in a typical 3D pixel or voxel in an MRI study, you have hundreds of thousands of neurons. And so basically your single number you get from each voxel is an average of the neural activity over hundreds of thousands of neurons. Okay, so the real miracle is that we ever see anything at all with this method. Yeah. Speak up.
Just to make sure I understand, so in that graph that you showed, what's the window that data after
What you typically do is you model that whole response, right? So in the experiment I'll describe in a second, we kind of bypass that whole thing by just flashing up a bunch of faces for 20 seconds. And so then, sure, the response is delayed by a few seconds, but if I have that whole 20 seconds, or maybe I leave out the first 6 seconds and take the rest of it, then I'll basically be fine. But if you want to do more precise things where you present individual trials more interleaved, then you basically model the response of each with a convolution of that function. Make sense? Okay. Okay, what else? So other dirty secrets of functional MRI. We can't measure absolute amounts of neural firing or metabolic activity, we just get some pretty arbitrary number, like 623, which is utterly meaningless except in comparison to some other task. So everything to do with functional MRI entails comparing the MRI response in one condition to another condition. Okay, and so it's also a key thing you need to think about when you read about MRI experiments is whenever anybody says, oh, brain region A is, responds in task Y, you should say compared to what, right? If what is staring at a dot doing nothing, then it's often not that surprising that that brain region is active when you do that thing, right? Okay. We also don't really know what kind of metabolic activity, what kind of neural activity underlies this MRI response. And we don't know if it's action potentials, as people tend to assume, or synaptic activity, or, I don't know, anything else, inhibition, right? And so in the limit, actually in one of the first papers it reported kind of sort of using PET, not functional MRI, functional MRI's precursor, there was sort of a face response in the bottom of the brain. And the argument was made that this is really ambiguous. Maybe that's the one part of the brain that's absolutely worst at face recognition. That's why the neurons there are firing so hard when they try to grapple with this stimulus, right? So there are many ambiguities in reasoning from blood flow to the function of a region. Okay. And very importantly, like other methods, single unit recording, most recording methods in neuroscience, they're just correlational. You change a stimulus, you change a task, you see what happens to the neural activity, but you don't know that that neural activity is causally related to behavior. Okay, and that's just a built-in limit. Was there another? Yeah.
When it would be fairly easy to establish correlation, like do an experiment with both PhDs brothers, where I see it correlation, and if they do, then there is some physiological correlation.
Yes, and people do that kind of thing. They do it more precisely. A famous paper by Nikos Logothetis, 10, 15 years ago, used monkeys where you could record from neurons in vivo electrodes right in the brain, and you could do functional MRI simultaneously, right? And then you could really say what exactly is the relationship between the kind of gold standard single unit activity and this MRI response. And his answer to that was the single unit activity is actually more correlated with, the MRI response is more correlated with synaptic activity than spiking activity, but actually it was correlated with both, and there's in subsequent studies, I would say subsequent studies are consistent with the idea that the main driver of the MRI response most of the time is spiking activity, though synaptic activity is probably also in the mix. Yeah.
When you say that this is a correlation but not a causal relation, why? Because it's no one when sees the face that it's firing.
Okay, let me state it more precisely. Yeah, I realized mid-sentence that I hadn't stated it right, and I thought nobody would notice, but good for you. There's a causal relationship. If you manipulate the stimulus, you, the experimenter, change the stimulus, and the neural activity changes. That's how you test causality, is manipulate something and measure the outcome. So that's causal. What's not typically causal in most MRI or physiology studies is a relationship between the neural activity and the behavior. So typically what you do is you have people doing a task with functional MRI. They're doing a task and you look at the neural activity. You don't know whether the ability to do the task was based on that neural activity. Yeah? Yeah.
So there hasn't been a way to initiate like a certain, like, I guess it's like initiating a certain neural response in the brain produces like the person who like, I don't know, like messes up their.
Yes, there is. We'll get there. There is.
Okay.
But all I mean is standard functional MRI can't do that. And opportunities to do that are extremely rare, but I'll show you one in a couple minutes. Okay. Absolutely. Absolutely. That's why it's important. Yep. Okay, so let me just go through super quick the most rudimentary kind of functional MRI experiment, just so it doesn't seem weird and mysterious, because it's actually really simple-minded. Okay, so here's one of the first studies I ever did. We pop subjects in the scanner. We show them a bunch of faces. We show them a bunch of objects. They're typically just lying there looking at the pictures, and we're scanning their brains. And we toggle between periods where there are lots of faces and periods when there are lots of objects over a 5.5-minute scan. And we typically take a full set of slices through the whole brain every 2 seconds. Okay, so it's like a movie with a frame every 2 seconds, showing neural activity. Everybody on board with this? So what you do then is you take these data and you say, for each pixel in the brain, you get a time course over that 5 minutes, and you just ask statistically, show the pixels in the brain where the signal was higher when the subject was looking at faces than when they were looking at objects. That's our first pass way to say, let's see if there's anything that's specialized for faces in the brain. Okay? And when you do that, in one of my colleagues 20 years ago, Randy Buckner at Harvard, I think this is Randy. This is a picture sliced through the bottom of his brain. This is the back of the head. Left and right are flipped here. And the statistics are telling us that this nice little tiny blob there is producing a higher signal when he was looking at faces than objects. Now, there's lots of ways to get nice little blobs by cheating. One way to tell that people aren't cheating is to ask to see the raw data that produced the blob. So here's mine. And you can see, just eyeballing it, that the signal, this is averaged over the voxels right there, that the signal is higher when he's looking at faces than when he's looking at objects. You can also see that the signal's higher when he's looking at objects than when he's staring at a dot at the points in between. Right? You can't tell people to just shut off their brain, or you can tell them, but they don't know how to do that. So for the visual system, one of the closest approximations people come up with is to put a dot there and have people fixate it so there's no eye movements. And so that's our baseline here. And so when I say this region responds selectively to, I don't mean exclusively. There is some non-zero response above this fixation baseline when you look at other things that aren't faces. Okay, everybody on board with that? Okay, so that's what a basic, like the most simple experiment looks like. But then if we want to ask this question that I've been carrying on about of how specialized is that region processing faces in particular? This question, this experiment doesn't answer that yet. Why not? That is, what else might you say about that response right here, other than that it's revealing a brain region that's specialized for face recognition in particular? You tell me. Give me alternative. This is what you do in science. You take some data and you don't believe it, and if you can come up with alternative accounts. So how else might we explain those data without postulating that there's a brain region specialized for face recognition? Like? Right? So for any of the kinds of stimuli you haven't tested, you don't know. So that's always true, and there's always going to be an infinite number of those. So this enterprise is never finished, right? It's always possible somebody will come along and say, you know what, I scanned aardvarks, and your face area likes aardvarks more than faces, and then the jig's up. Could always happen. Could happen to me tomorrow. I was like, okay, jig's up, right? And so, yeah, that could be true for bodies. It could be true for other things. But let's think about why bodies in particular. So you might say, for example, okay, it's not about faces in particular. It's about, human body parts, maybe, right? Absolutely. It's a totally viable hypothesis given these data. What else? Yeah. Yeah, maybe round things. Yeah, absolutely. What else? How else might you account for these data? Or what else might that thing be doing given these data? Yeah, skin color. Yeah, it could be doing any number of different things, right? Yeah. Totally, totally. I mean, talk about apples, and it's worse than oranges, this contrast, right? Apples and oranges would be much tighter contrast, yeah. I'll show you one shortly. Okay, so my point in doing this is just, it's not rocket science. Like the way, if you really want to make a claim about specialization of a region of the brain, you need to engage in this kind of enterprise. And there are usually many, many alternative accounts. And the only way to really nail it is to take the main ones seriously and test them. So I'll just give you one example of how we did that early on in our first paper. Oh, first of all, you see this thing in every subject you test. That region will be in slightly different locations, but it's always back there on the bottom of the back of the right hemisphere and the lateral side of the fusiform gyrus. Okay, so experiment we did was we said, okay, maybe it responds to anything human or any body part, she mentioned, or anything people pay attention to, right? We're social primates. We're interested in faces than we are in ears of corn and crabs and telephonins and whatever else we had in there, right? Or anything with curvy edges related to your hypothesis, right? So one of the tests we did was to do a new study in the same subjects. We used slightly different faces just to see if it generalized to 3/4 views, to make sure it was just hair. And we used hands as our control condition, saying if it's really faces in particular, the response should be higher to faces than hands. Further, to address this attention idea, we said, well, let's make subjects pay even more attention to the other thing that isn't faces. And we did that by adding on a task, where the task is whenever two consecutive images are identical, you have to press a button, okay? And that task is pretty easy with the faces, and by design was killer hard with the hands. So we forced subjects to pay more attention to the hands, biasing against our hypothesis. And what we get was the very same blob responds more to the faces than hands. And here's the raw data. And so that's just an example of how you can do other experiments to really drill in and say, no, it's not anything human or any body part or anything we pay more attention to or anything with curvy edges, okay? It leaves many other hypotheses open. It's just to show you how you can engage in this kind of research program. Make sense? Yeah. Yeah.
Is this like a condition of like
Yes. Yes. You can do that, and I've done that. Other people are doing it. I was just Skyping with one of my former postdocs yesterday. He's doing it in yet another kind of very clever way. And the basic answer in all of those cases is the signal just kind of, you take a full face and you deconstruct it in any way you can think, like put ovals in the same position over the parts, or get rid of the outside contours, or keep the individual parts intact but move them around into the wrong place of the face. All of those manipulations will lower the response but still leave it higher than pretty much any object. But that's also very ambiguous because it doesn't tell us whether all of the neurons in that region are responding with that graded way or if some neurons are responding to each of them. And so there are many ambiguities there. Yeah. Okay. So basically, we've been at this enterprise for a long time, and there are lots and lots of conditions that have been tested, only a few of them shown here. I'll just point out one other. If you haven't seen this image before, raise your hand if you can tell what it is. Everybody see it right there? This guy, can you tell what that is? Raise your hand. Okay. Raise your hand if you can tell what that one is on the top. Come on, you guys. Okay, if you haven't seen it yet, don't worry, it's a little subtle. There's nothing wrong with you. It's a profile view of a face, eyes, nose, mouth. Okay, and the important thing is this is the same image, it's just upside down. And this recapitulates a discovery that was made actually in this department by Robert Yin in his PhD in 1969 using the very high-tech equipment of paper and pencil and a stopwatch. And Robert Yin showed that face recognition is severely disrupted when you present faces upside down. And that inversion cost is much greater for faces than for any other category of stimuli, landscapes, objects, words, whatever. And Yin inferred from his elegant but very low-tech data that there may be specialized brain machinery for face recognition. Isn't that cool? Anyway, so we're using that method here as a way to control for the visual properties of the stimulus, and we show that when subjects see the face in that image, the response is much higher than when they don't, even though it's the same image upside down. That enables us to rule out a whole host of alternative accounts, like, oh, maybe it's the particular spatial frequency composition of your image, blah, 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 right? No, it's not, right? Okay, so all of that to say, at this point, it's very clear that this thing prefers faces. Using this kind of method, basically coming up with a hypothesis and testing to try to rule out alternatives. This is me with some of my special bits shown. And so the main picture I'm trying to sell to you guys is that the human brain contains a lot of functionally distinctive regions, many of them highly specialized for solving very specific problems. Okay. And I want to quickly mention a few clarifications of what I mean by this claim about functional specificity. First of all, I don't think it applies to all of the cortex. If there's time, I'll later talk about these white regions. The white regions seem to be engaged whenever you do anything difficult at all. They're like the opposite of these specialized regions. They're very, very general purpose regions. Nobody really understands what they do. I think they're in some ways more mysterious and more fascinating than all the specialized ones. Anyway, so not true of all of cortex. And second of all, for the regions that are functionally specific, the claim is simply, in normal adults, this region does a very specific thing. That's the only claim. Do not confuse that claim, as my colleagues do, with claims that it's innate. That's a really interesting question, a totally different question. You'd be shocked how many of my colleagues don't understand the difference between those two claims. With the claim that it's unique, like if I say here's a region of the brain that's specialized for face recognition, it doesn't mean there aren't 6 others. There might be 6 others. There's in fact at least a few. And if there's a few, that's cool, that's great, and no way does that undercut the specificity of this one. In some ways it makes it more interesting. And third, claiming that a brain region does something very specific in no way means that that region acts alone. No region of the brain acts alone. Every region of the brain needs to get input from other areas, so it has something to process, and it has to send output to other areas. Otherwise, there's no point in doing that computation if it's not telling its friends what it found out. Yeah? So everything is connected. Nonetheless, despite that connectivity, there are bits that seem to do very specific things. Okay. So as I said, it's an old idea. I'm not going to get into the controversy. I should say, though, that this view that I'm putting forth, and part of the reason I take the time to show you the evidence, is this is considered controversial. Frankly, I have no idea why it's considered controversial, because I think the evidence is actually very strong. But I'm giving you a slightly fringy view here. I'm giving you that view because I think it's right. Yeah. Oh, that's a great question. It does, usually. But remember that I said MRI can only show you differences between conditions. Right? So for example, part of the reason that we started off contrasting looking at faces versus looking at objects, like notice we could have just said, okay, let's show people faces versus having them stare at a dot. You can do that, and you will get that region. But you will also get all of primary visual cortex, because all of primary visual cortex likes visual input and will turn on like mad, right? And so part of the reason we chose that contrast is the question was, over and above vision in general, is there a bit that's specialized for face recognition, that engages in face recognition more than it engages in other kinds of visual shape-related tasks? See what I mean? So there are other regions before and after that respond to both. So if there are other regions that are specifically processing the output of the face area, you will also see those in the same contrast. Or you might. There's lots of ways to not see things that are actually there. But you have a shot at it, at least. Okay, so I think this is important because it's basically giving us a rough sketch of our minds and who we are, and I think that's fun. Let's see, what am I gonna, I'm taking way longer than, yeah.
Just about data again.
Yeah.
Is it always correlated? You see people are against or the activity in this area is correlated?
It's a good question. There's so many people who have looked for that kind of thing. I'm sure that there's literature that I'm not thinking of, but I don't know of any cases where people, I mean, like the strong test would be, can you show a case where there's a very strong response there and people are not experiencing a face, or vice versa? They're experiencing a face and not getting a strong response there. I don't know of any. I would consider such data to be strong evidence against the story I'm telling you right now. And I don't know of such. Run into such, send me an email. So let's talk. I mean, I think there are many cases that are on the border where you can see a face or not. My prediction would be if you have a minimal image that contains a face, if the person's seeing the face in it, you will get a near maximal response here. And just below that, it will be very similar to objects. Actually, slight evidence against this. Pavan has a paper where he took some false positives from some computer vision algorithms, where the algorithm saw a face and people did not. And I think the claim is that you get an intermediate response, but I forget the details. Yeah. Okay. I think what I'm going to, I have a cool, I know people are going to get hungry, and I'm aware of that. I will too. So you're safe. But I think what I'm going to do is just mention what I consider to be the strongest challenge to the view that I'm putting forth here, and then show you a cool video that at least somewhat addresses that. And then we'll come back and figure out which of the things to talk about. Does that make sense? Okay. So I think there's lots of evidence for this view that I'm putting forth, and there's a lot of what I take to be just wrong-headed criticism that I won't waste your time with. But there's one really smart criticism, which I take very seriously. And that is that if you look at this picture that I showed you, first of all, you can see that there is, in fact, some response to non-faces, right? And second of all, this whole line of argument comes from Jim here and many other people who've kind of taken this on, but it's really his thinking that's behind this. He wrote a very influential article in 2001 where he pointed out that even if you look just within the face area, there's a fine-grained pattern of response across the voxels in that region, right? Roughly 100 voxels. And that fine-grained pattern of response differs systematically for different categories, right? So for example, if you scan people while they look at, say, chairs and cars, and this is a schematic, imagine the fusiform face area flattened out, and this is the response of each voxel. It's just fake data. If you do this and you find that systematically those patterns differ, well, then the face area contains information about the discrimination between chairs and cars, right? And that's not faces, okay? And there's lots of ways to show this. You can use simple correlation methods, as Haxby did. You can train up linear support machines, and you can do all kinds of other fancy things that you guys could invent better than I could, all of which are ways to look at that pattern and read out different information, okay? And so this enterprise has been going on for a while. When Haxby first reported this, I said, well, you didn't define your fusiform face area right, and when I define it right, I don't see that, and then it went back and forth, and eventually I said, whoops, actually now at higher resolution, I do see it, you're right. And then he followed up with a paper saying, yes, but actually, that information's really weak. So we met right in the middle. It's there, it's significant, it's weak, but it's there. And to the extent that we think the brain is processing information, that's the assumption of our whole field, then we should care not just about how strong the response is, we should care even more about what information is in that region. And so this is a very important challenge. If there's information about other things beyond faces, who cares what the mean magnitude of response is? Okay, everybody feel the force of this? Is a serious concern? Okay. This is sometimes called multiple voxel pattern analysis, this business of looking at the fine-grained patterns, and it's gone wild. There's millions of variations of it, and it's all very exciting and cool. It's very important because we do want to know not just what activates a region, but what it represents. And this is our main method.
Could it be the multi-face feature of the cloud?
It could be any number of things. Any number of things. So I'm trying to get you guys to see that this is a serious challenge, but not that there aren't ways of responding. Absolutely. Absolutely. All you'd have to do is say, yeah, the car is more face-like than a chair is. And then you would be able to decode this, right? And so then the real question is not what can we, the scientists, decode from that region, but what is the rest of the brain reading out from that region, right? So this is where the causal tests become important. With functional MRI, we can't tell that. We can use fancier and fancier math to read out more and more impressive things out of a pattern of response across voxels, but then we have no idea if that's our prowess as neuroscientists reading that stuff out, or if that's actually what the rest of the brain is reading out of there as well, okay? So mostly this is a big problem actually also in systems neuroscience, but very in human cognitive neuroscience. So everybody's in the same boat. But in systems neuroscience, if you have a monkey, you can at least disrupt that region and look at behavior more often, and so you have more levers to go at. With humans, we have a tough time, but I will show you a rare opportunity that I had. Did I give you this paper? I don't think I did. It just got accepted yesterday or something like that. where we had an opportunity to test a patient with electrodes in his brain being mapped out before neurosurgery, where the electrodes were placed right along the bottom surface of the brain, right in the regions that I care about. And the neurosurgeons decided for medical reasons to electrically stimulate regions on the bottom of the brain and ask the patient what happened. This is a way to test the causal role of those regions in perception. Okay? So I'm going to show you a video of that in a second, but let me give you a little more background. Just for comparison, that's the bottom of my brain there. The red bits are my face selective bits in the right hemisphere and the left hemisphere. And this is the patient, the bottom of his brain, and each of those little things is an electrode that's been placed on the surface of the brain. This is very extreme neurosurgery. You hope this never happens to you or anyone you care about. But if you have intractable epilepsy, as this patient had, that was not treatable by drugs, this was, okay. Okay, so here's the patient. You'll meet him in a second. This was done in Japan. He's speaking Japanese, but you'll get there's subtitles, and you'll get the gist of what he's saying anyway. Okay, so the first thing we did was to record the responses of each of those electrodes to a bunch of different stimulus conditions. So we I learned about that this was going to happen about 36 hours before it happened, and so everybody in my lab dropped everything to make stimuli, and send them to Japan. Okay, so we presented stimuli that are bodies, faces, digit strings, hiragana and kanji, two different kinds of Japanese characters, line drawings of objects, and photographs of objects. And we presented them both in color and grayscale. So this is not the causal test of the relationship to behavior. This is just mapping out the selectivity using electrodes, because this is a much more direct measure of neural activity than functional MRI. We're not going by way of the blood flow response. We're direct recording electrical responses on the surface of the brain. Okay. And so we can ask, what happens to that electrode right there that I've circled in red, which looks like it should be right around in the face area? Well, this is what happens. A humongous response to faces, a teeny tiny response to bodies, body parts and faces, and pretty much no response to anything else. Now, one thing you see here is that the selectivity of this response is much greater than I showed you with functional MRI. I think that's because we're actually recording the real deal with more precise methods that aren't blurred by blood flow that smears around in the brain and that are showing us a region that is actually really for faces, right? I suspect that's true in all our functional MRI data as well. It just gets blurred by the method. Yeah. So that's a nice reality check. Right next door, I mentioned briefly that right next to the face preferring region is a color preferring region. And so right next door, that electrode does this. Okay, now what these bars mean is the outer bar is a response to that condition when it's in color, and the inner bar is when it's presented in grayscale. And so you can see that across all the different stimulus categories, that electrode responds more to the color version than the grayscale version. So it's got some preference for color. Now, you'll also notice that the preference for color in the color electrode is weaker than the preference for faces in the face electrode. Okay? So my terminology is that's face selective and this is color preferring. It's a fine point to avoid hassle from colleagues. You guys don't care about that. Anyway, just note that it's slightly weaker. Okay, so far, that's nice, but all here is replicate with electrical recording what we found previously with functional MRI. But that's great. As a friend of mine says, I'm a replication junkie. We don't have enough of this in science, and especially when you replicate with a totally different method, indeed a better method, it's a fabulous thing. Okay? Nonetheless, the real question I started off with is what's the causal role of these regions? So now that we've established the selectivities of these electrodes, now we can ask what happens when they're electrically stimulated. Okay? And so I'll show you. We'll start with electrical stimulation of the face selective electrode, that electrode right over there. Now realize this doesn't know there's a face selective region, has no idea which electrode is being stimulated, and is just asked to report what he experiences. Now I'll say that we weren't the first to try this. There's several papers that both record electrical activity and also stimulate regions here and nearby that have already shown the causal role of face selective sites in face perception. Okay, that's really important, and you'll see it in my data too in a second. But my real agenda, since that had sort of been shown, was to address this challenge from Jim Haxby and others. If there's information in the fine-grained pattern of response across that region about things that aren't faces, And surely if we hit that region with a big sledgehammer by putting in a big current right there, that should disrupt that fine-grained pattern. And therefore, it should mess up the perception of other things that aren't faces. That was the key question, okay? So I'll show you what happens when this guy is stimulated while looking at faces and what happens when he's stimulated there while looking at things that aren't faces, all right? Okay, here we go. Okay, so he's looking at a face and he's getting stimulated right on the face selective electrode, and it says, and he's completely changed.
全く違う人なんだ。違う人になります。どんな感じの人でしょう。髪型は変わってないんですけど、ね、言葉であれするですね。もう一回やって。もう一回。One more time.
Such a great subject. He's just trying to get it right, you know? Okay, so he's getting zapped again right there.
この部分だけなんかアニメでできそうな感じのキャラクターの顔みたいに変わったんですけど、口はそんなに変わってなかったんですけど、鼻目こんな顔でした。
Okay, so now he's looking at a box, okay? And he's going to get in the same electrode right there.
No change.
But
最初の1秒ぐらい、始まって1秒ぐらいがなんか横になんか目口って感じでちょっと一瞬見えたんですけど、これ何かなと思ってたら気が付いたらもうこの箱にしか見えてないっていうか。
Okay, so then he's looking at a round object. We thought maybe he'd be more likely to with that.
なんですか、よくまたさっきみたいに横にその目と口って感じでちょっと。
Okay, this is a kanji character on a. It's foreshortened. You'll see it in a second.
More, please.
One more time.
Yeah, やっぱり顔に見えました。目、口らへんって感じ。
So the point is, one, that region is causally involved in face perception. It doesn't just turn on when you look at faces for some epiphenomenal reason. It's actually causally involved in that if you directly reach in and activate those neurons, the person sees a face. But at least as importantly, even though Haxby and others, including me, have shown that that region contains fine-grained information about other things beyond faces, if you read it out, you can read it out. When you ask him how stimulation of that region affects perception of something that isn't a face, it doesn't do anything other than stick a face on top. And so, to me, this is pretty strong evidence that even if there is fine-grained information in there, it's probably not being read out, or it's not playing a major role because we radically disrupted it, and apparently nothing happened to the percept of the non-face object. Yeah? Speak up. Yeah, that would be very interesting. It'd be interesting to do that and to have him look at a blank page. We didn't have time to do any of that. So, you know, these experiments, you know, I'll never get another shot at this. Like these experiments are really rare and this is like, you know, pretty much never happens. And when it happens, there's just a tiny little window. We have, 40 minutes of data total, that's it. So yeah, there are lots. I really wish I had been there to ask him in more detail, like really tell me about the shape of the box. Tell me about the shape of the ball. Did anything about the box or the ball or the kanji itself change? As far as I can tell from the transcript, and we have, I think, 8 stimulations on, I forget how many, we have quite a few. As far as I can tell, nothing about the non-face objects changed. They're just face things on top. But it would be nice to interrogate further. Yeah, the face was sideways. I have no idea why. I think that's very interesting, but I don't know. Yeah. Okay, so I'm almost done and we'll have a break, but let me just show you something else first. So that's what happens when you stimulate that region. It shows the causal role of that region in face perception, and I would argue the pretty much exclusive causal role of that region in face perception. Notice he could have reported anything else. He was asked to report anything that changed, and he didn't report anything else other than seeing a face. But we're social primates. We see faces in clouds. We see faces all the time when they're not there. Maybe any damn thing you do to the brain makes you see a face. Maybe you see a face, right? So let's ask what happens in other electrodes. And in fact, let's ask what happens when you stimulate right next door in this electrode that responds preferentially to color. Okay, here's what happens. He's looking at the box, getting stimulated. No, it's kind of an old box, so there's texture, but there's no image on it.
この箱の真ん中見てたら、
Is that crazy or what?
この半分ぐらいがなんか虹色になんか光ったりしてますね。この真ん中見てたら、まあ、ボール見てたらもやっぱ先が明らかに幅広い感じ。なんかちょっと虹色に点滅してるんですけど。先生のやっぱ鼻あたりに目線見てたらやっぱこちらの顔半分が
That's the kanji.
まあ同じような感じで半分がカラフル。
So that shows that conversely, this region is causally involved in color perception. And importantly, it's right next door. It's like a centimeter away from the face selective electrode. And that also shows you that it's not just that if you stimulate anywhere in the vicinity, you get the same thing. Instead, what we see here is a fine-grained functional organization that corresponds to what was seen before with functional MRI, and where we now have the ability to show the causal role plus the specificity of that causal role. Yeah?
Do you have any idea like why it's only half?
Yes. Yes, because each side of the brain, especially in perceptual regions, responds to the opposite side of space. He's stimulated in his right fusiform gyrus. That's why he sees color on the left.